if that's okay. All right. Thank you, everybody, for being here this afternoon. I'm Kurt Hankinson. I'm a faculty member in orthopedic surgery, and we've been in our group doing a lot of single cell and spatial of joints and bones, and asked Robert Tower to come because he's really the person who has led that field during his postdoctoral work and as an assistant professor over the last decade or so. So first doing single cell of bone marrow, other tissues, and then into the spatial transcriptomic area. So Robert and I have been collaborators for a number of years. He grew up in Canada, so he's Canadian. Went to the University of Alberta, so he's from Alberta, Canada. Did a BS and a master's there before doing a PhD in Germany, and his PhD in Germany was focused on imaging of bone. So he did some really elegant work on targeting of imaging agents to bone. And then he moved to KU Leuven, so he moved to Belgium and joined the laboratory of Christa Mays, who was one of the leaders in the field of identifying progenitor cells in bone. And he had a very prestigious Flemish scholarship to go there, an FWO postdoctoral fellowship. He was there for a few years and then moved to the University of Pennsylvania to work with Ling Chen. And Ling at that time was just starting to enter the field of single cell sequencing of bone, and Robert really led those efforts and had a number of very high profile papers that were published with Ling over many years. And then he moved to Tom Clemens' lab at Johns Hopkins before taking his position at the University of Texas Southwest, so UT Southwest, where he's an assistant professor. And he's studying all aspects of bone using these techniques that he's here to discuss. And so I'll end it there. Thanks so much for being here.
Yeah, thank you very much. Everyone can hear me all right? Yeah, I wasn't really too sure of the format that these normally go. So this was more of like my journey in using spatial in the bone field. And so it's several projects, so I apologize if it feels a little bit disjointed and that there's a lot coming at you. I don't mind this being interactive. I think it was designed more to be like, how did you make that work, or how does that play out, rather than let me ask you about the biology. But in the end, I think this is nothing more than a tool, right? And it's what do we do with it that's really going to make the difference between, you know, just I did single cell, here's my UMAP, versus I've discovered something that's actually going to be relevant to people. So let's see. Maybe we could probably skip this, but everyone knows the basics. We started off with microarrays and bulk seq, and basically you're taking a heterogeneous tissue, extracting all the RNA, and looking for changes in gene expression. The problem was always going to be that if you see a change, is that because you have the exact same cell types that actually have up or down regulated the number of transcripts, or is that because you've had an entrance or exit of a certain type of cell within the tissue? And so this was overcome when we started to do single cell RNA sequencing, where now you're dissociating those cells and looking at the transcriptional profile of each one individually. And so this has been a huge advancement in terms of now we can look at both what cell types are expressing our genes of interest, as well as within that cell type, how does my perturbation or drug treatment or knockout change the overall expression? And so while we still widely use this today, the problem has always been that you require this dissociation into this single cell suspension, right? And we know that this is not what's happening in vivo. These cells are not floating around in space. They are constantly interacting with the matrix. They're interacting with the neighboring cells. They're exposed to signaling gradients, availability of metabolites, and things like this. And so this is kind of, I guess, the next evolution of now we're going to try and look at transcriptional profiling of hopefully single cells, but actually in the context of their in vivo nature. So most of the work that I've done so far is going to be focused on Visium. So just in case anyone's not so familiar, it's as well gone through several iterations, but the basic idea is you're going to take a histology section and place it down into their overpriced slide, or another slide that's also a little bit overpriced, and transfer it to their even more overpriced slide. And you're going to try and capture either directly the mRNA in the context of fresh frozen, or you're going to capture probes that have bound to your mRNA molecules along the slide surface. Now, these oligos along the slide surface have this spatial barcode embedded into them. So it's a series of nucleotides, and we're going to use this. And so when you sequence your sample, it's going to read this message, and it's going to say, this is the exact two-dimensional position that this piece of mRNA came from. And when you combine that with your histology section that we do H&E imaging on, this is what's going to give you both the tissue morphology as well as the gene expression that's occurring at these two-dimensional positions. So I mentioned this is more of a journey through my experience of using spatial in a variety of projects. And so one of the first ones that we started to look at is in the context of calvarial suture development. And so the calvaria, or your skull, starts off as these floating islands of bone that are connected by this undifferentiated mesenchymal tissue. And so these bony islands, if you will, are going to continually expand in order to kind of facilitate skull growth, while at the same time, this undifferentiated tissue is going to be constantly providing them with cells and maintain this kind of loose gap in between. And this is what lets your skull grow as your brain develops. And so if you have this disruption of stem cell expansion and osteogenic differentiation, you have this misbalance of suture being maintained and the bony plates will fuse. This causes diseases like cranial synostosis that cause severe developmental issues. And so in the lab of Tom Clemens, we were looking at the role of sensory nerves shown in red and how they're going to regulate this calvarial suture. And you can see that not only is it highly innervated, but it's recruited through the robust expression of this neurotrophin nerve growth factor shown in green. And when you look at this mineralization rate, so this is in utero dynamic histomorphometry where you inject dyes and they're going to label surfaces that are mineralizing. And so you can see kind of this yellowy green color is going to be where the bones kind of initially started when we did our first injection. And when you do a second injection, you see this red front. And so this is how much these bones have grown. And when you look at the distance between these probes, what you notice is when you get rid of this sensory nerves, when you block via this TrkA receptor, you can see that this rate of mineralization is increasing. So the problem has always been that the sutures are very small, and they contain every cell type from the most undifferentiated stem cell all the way to the most terminally differentiated osteoblast within a very confined space. And so we ended up doing spatial transcriptomics on this, and this is the first version, so my resolution's not amazing, and not surprisingly, because of this size of the tissue, you can see that one of the first limitations we first came across is that there's very few spots that are encompassing this calvarial suture tissue. And so this has also been a little bit of a journey of how do you start to overcome a lot of the limitations that were present and really are still present in a lot of the spatial platforms. And we started looking at some pathways, and this one's focusing on BMP signaling. And you can look with the violin plots, overall levels are not so super exciting, but we noticed that there seems to be not just about BMP activation, but where this was occurring. And so to overcome a few of these obstacles, and I'm going to come back to this again and again because we really like it, is this idea that we created called spatial time. And if you're thinking that sounds familiar, that's because we stole the idea from single cell pseudotime, where they align their single cell data from stem cell on the left all the way up to mature cell on the right. Well, we do this exact same thing in space with our stem cells, which are right down the middle, and our differentiated cells along the outer edges. And so what this is letting you do is measuring how far away each spatial spot is from this central midline region where our stem cells are. But this also lets you start pooling together multiple samples together. And so every suture is slightly different shaped. It's going to be bigger or smaller. But by doing this, it allows us to put every single spot on a linear axis of how far away am I from that midline suture region. And this is going to give you more statistical power to start to investigate some of these lower expressed genes. And when you look at how these BMP activators and inhibitors are expressed, and you can start to see little shifts, but it's probably easier to see when you graph it. Normally, in the black curve, you have this kind of wave of BMP activation as they leave the stem cell and enter into this osteogenic differentiation. And this is what's going to allow this nicely coordinated maintaining stem cell plasticity while also allowing your kind of skull bones to continue to expand. If you get rid of the TrkA signaling in this case, you can see that this BMP wave is dramatically shifted all the way into this central midline region that really shouldn't be exposed to these types of signals. And when you stain, you can see that while normally BMP activation is heavily enriched along these osteogenic fronts where the bone's going to form, while minimal present within that midline suture, in the TrkA mutants, you can see BMP activation kind of scattered throughout. And so this is showing us that sensory nerves help maintain suture patency during development by spatially regulating this BMP signaling. So it was around this time that I started to make my move over to Dallas, and I started working with a colleague on digitip regeneration. And so for those of you not familiar, there are animals out there like axolotls, and I think I talked with one person in the audience today about it, that you can cut off an entire arm and they will regrow just fine. In fact, we're going to get to axolotls at the end of the talk. By comparison, mammals are far inferior in terms of our ability to regenerate, right? We have the ability to regenerate the distal 1/3 of our third phalangeal element, which equates to about this much of your fingertip, right? So what happens when you amputate, you'll actually have this histolytic event that'll chew back some of the bone a little bit further, and then it'll actually grow out to form and reform this kind of overall digit shape. Now, it doesn't look super amazing compared to regrowing a whole arm, but if you think about it, compared to a lot of the other injuries, we usually get signaling cues from multiple directions, right? If you fracture a bone, you have this broken piece and this broken piece saying grow towards each other and fill the gap. In this case, you're amputating and you're telling it go forth into outer space, essentially, right? And so somehow there is a signal that's telling them these cells have to grow. They have to grow in that direction. They have to form and pattern in this way. And so in this project, this was collaboration with Mimi Samarkku, who's at Mayo Clinic now. She was interested in the effects of aging. And it's not surprising that aging is bad for everything. And indeed, it's worse for digitip regeneration. And when you quantify the micro-CT, you can see that overall these digit tips are not only forming less bone, but the bone is really chaotic and messy. Now, the problem in that field, again, similar to the calvarial suture, has always been this critical structure right here. This is known as the blastema, and this is what's making the regenerative magic happen. This is going to be a mixed bag of stem cell and progenitor cells coming from all different tissue types. And this is going to encode and provide the cells and the mechanism to regrow the entire digit out. But if you don't have scale bars, so it's going to be hard to imagine, but you can imagine how small the very distal tip of 1/3 of the mouse finger is. So it's to the point where even just getting the first single cell data sets coming from these samples only happened a few years ago. So this is one area that I also think that spatial is really well suited to do, and that you don't have this reliance on a certain level of input of tissue. If you can get a tissue section, then you can run spatial on it. Now, I should say, for obvious financial reasons, we did not put a single digit tip into their reaction window and run it. We jam-packed that thing in there to get our money's worth. But in the end, as long as you can visualize it, then you can analyze it. I think the other benefit that spatial is going to bring to this is we've all done single cell and we're running our differentially expressed genes and we're trying to find a marker to stain my population of interest. And usually when you pick one out and you go to stain it, you're always a little bit disappointed of, oh, it was so good in my UMAP, but then when I stained it, it was a little bit everywhere, right? And I think this comes to the fact that when we run single cell, it's really hard to get every single cell type that's going to be present in your tissue sample. Right? Some cells are notoriously fragile, and as soon as you try to dissociate, they die. Some of them are so embedded in matrix, it doesn't matter how much digestion you do, it'll never come out. And so some of the times we have to micro-dissect out tissue like skin. It's because we don't want all the fibroblasts to overwhelm the cell types of interest. So we're always biasing ourselves in terms of what's actually going to be present in our single cell data set, and we're looking for genes that are enriched in one cluster versus everything else that's present, but you're missing a lot of information. But with spatial, we have all the tissues present that are going to be looked at. And so in our experience, this tends to map up better in terms of finding a unique marker that labels the structure. And so the first thing we did was basically find out what's unique about the blastema versus everything else around it. And how few genes can we work our way down to before we can basically not successfully label it? And so we developed this kind of blastema gene signature, but then we came back to our favorite spatial time, in this case, so improving it. Instead of picking a spatial spot, now our reference tissue is going to be the bone stump. And we were looking for signals that basically change as you move along this axis from amputated region all the way out to the distal tip of the blastema. And one of the pathways we had picked out is a lot of genes related to metabolic regulation. And we looked at this, the aging mice tended to be more enriched in expression of genes associated with glycolysis when you're near the proximal end or near the amputated region, and more enriched for glycolytic genes as you move, more related to oxidative phosphorylation genes as you move more distal. And so this is also something that, although this is not looking at metabolites or metabolic flux, this enrichment for metabolic genes is also something that's really hard to do in a lot of different other assays. And we went in and tried to validate. We can see that in these aged mice, they had an increased bioenergetic demand. And I think the moral of this, not to go into too much detail, but there was an issue in terms of generating enough progenitor cells to fill in that blastema to give critical mass. And so these cells were having to work much harder in the aged digits because they didn't have as many of these starting building blocks. And actually, by supplementing them with this increased energetic needs, we were able to rescue this aging-induced impaired regeneration. So in this case, we use spatial transcriptomics to not only define what a blastema is, which till that point was really just more of a morphological structure, but also identified these changes in metabolism that were driving this inherent disorder. So this was the point where I really started to get more into it, and we started to see what was really possible. And it was around this time that they started to bring in this FFPE version of spatial transcriptomics. So everything up until this point was all fresh frozen, and we were able to overcome the fact that there is some mineralized tissue by either relying on like neonates or by optimizing the sectioning parameters. But now we really want to start looking at the context of an adult bone. And so to give a brief overview, you have a nice cortical shell, and inside is going to be your marrow cavity that's going to be also containing trabecular bone that's all about mechanical support. But what's interesting in bone is that this is constantly being turned over. So you can imagine how much effort it is to turn over your entire skeleton. But without this, you're going to slowly develop these deficiencies, and this is what's going to lead to issues down the road. And so probably the most common disease that we're all aware of, normally what you end up having is a coordinated action of these bone-resorbing osteoclasts and these bone-forming osteoblasts, right? And you need to maintain these at an equal balance. And what happens as you age is you tend to resorb too much bone and not form enough. And this leads to progressive bone loss. And you can imagine that at some point, your bones are going to get so weak that fractures will happen. And so I mentioned this is really driven by, at least one side of the equation is driven by osteoblasts. And so these osteoblasts, they start off as these stem or progenitor cells, however you like to call them. And they have a choice to become either an adipocyte or an osteoblast. And so we were interested in what were the processes and pathways that are driving this overall decision-making process. And again, this was just the first version of this FFPE blocks, which means we're still stuck with that 55 micron resolution. And the problem in the bone marrow has always been, well, I only care about this. There's a whole other world that's happening in the exact same region, right? The entire hematopoietic lineage is going to be present within this exact same bone marrow in combination with nerves and vasculature and smooth muscle cells. And so how do we start to use this spatial to try to unravel these two complex processes? And so for this project, we wanted to tackle 4 questions. One of them is, what are these skeletal stem and progenitor cells? And this is going to be in relation specifically to some questions that were raised from single cell data that came earlier. We want to know, what are the cell types that are present within this stem cell niche that are helping to promote and regulate their function? How are they talking to each other? And finally get at this idea of these microdomains that are present within the bone. And so our first ever run of FFPE tissue, we just processed them like we normally would. And honestly, the results were not as amazing as we'd hoped. And that is because everything you do to remove minerals so that you can section a bone also basically destroys the RNA. And so this is an area of ongoing work to try to optimize this, but in at least our first batch, we were able to section it, and while the depth wasn't amazing, we still were getting genes expressed where you'd expect, with more osteogenic genes expressed in the cortical and trabecular bone, and then more hematopoietic type genes expressed in the bone marrow. And so we come back to the stem cell niche, and we know this is critical in all forms of cell biology or stem cell biology. But again, the problem has always been in the context of a Visium 55 micron spot, you're basically getting a giant blob encompassing this entire dynamic, unique structure, right? But luckily, there's an abundant resource out there that contains these cells at a single cell resolution, and that is, of course, single cell. And even at this point, there was multiple groups that had published on a variety of models, and so we essentially combined all of the data we could find into a single kind of bone marrow atlas, although I don't really like the term so much, to compass all the major cell types in the bone. And I'll also point out as we move forward that it's really important when you're trying to do what we're going to do coming up that you get all the cell types present. And this also comes back to this idea of when we do single cell, we're not really getting everything. And especially in the context of this one, you'll notice a lot of samples that are coming from the bone marrow, they've either sorted out or removed all the red blood cells. Now, while I don't have a strong love for red blood cells, I'm sorry, they actually are fairly prominent in the bone marrow, right? And so you can imagine we're trying to build a puzzle, and I've just removed 60% of the pieces that are actually present. Yeah, 60, probably more, right? So now when you're trying to reconstruct this, what you end up doing is like what my child does, is he takes a piece that looks kind of similar and he starts smashing it to try and make the pieces fit, right? So when you're going to do these kind of convolution that we're going to talk about, it's really important that you actually start with all the pieces that are present. Otherwise, disaster will happen. But anyways, how we're going to overcome this issue of our giant blob encompassing an entire or multiple niches is spatial deconvolution. And so this is a way that we know that these spatial spots encompass numerous cell types. And we are going to use a transcriptional profile from our single cell to try to unravel what cell types are present within each spatial spot. And a good example we heard earlier is this idea of spectral unmixing. It's the same idea, but transcriptionally happening. And so we use a series of packages that had been developed. Anyone who's done single cell analysis will know of SURAT, but also CellTrek and Cell2 location had all been published kind of around the same time. And these are all different techniques with the exact same purpose. So computationally, I'm going to be honest, I don't really understand how they work, but I know that they work in slightly different manners to all take a spatial data set and a single cell data set and tell me within that spatial spot who's present. And so when we start to look at our bone spatial and we start to break it apart, you can see on the left-hand side, this is our cortical bone. So cortical bone should be primarily made up of these mesenchymal lineage cells in red. And so SURAT is doing a very good job of saying, you know what, most of these spots are going to be containing nothing but a mesenchymal lineage cell. Unfortunately, when you look at the bone marrow, it's also saying half of those spots are containing those exact same mesenchymal lineage cells, which we know is an overestimation based on a lot of histology and transgenic mouse work. If we look at Cell2 location, now we can see that the mesenchymal lineage cells have disappeared from the marrow. We're overrepresenting in a lot of red blood cells and immune cells like you'd expect, but for some reason it thinks that the majority of this cortical bone is now made of smooth muscle cells, which we also know is incorrect. And then we actually have CellTrek that kind of performs intermediate in most of the parameters, but neither one of them was really optimal. And I'm going to admit that it probably had something to do with the poor quality of the spatial data. The packages, I think, were probably okay. But what we ended up doing is create a pipeline where we merge all three of these approaches to try and pull out the best of each and eliminate the weaknesses of them as well. When we do this kind of combined approach, what you end up getting is a little bit more of a distribution of cell types that you would anticipate just based off of our morphology and our known science from the background, right? So cortical bone, again, primarily mesenchymal lineage with some vessels moving through it, bone marrow, a lot of red blood cells with a lot of immune, but very little mesenchymal lineage. So we feel like now we're at least a little bit more accurate in terms of taking these giant spots and piecing them down and saying which cell types are present in which areas. And so the first question we wanted to ask is, there was a series of papers that were coming out and describing multiple potential stem cells that are present in the bone marrow. And forget about what they actually named them. What I think they actually are, we have these P-alpha-S cells that are enriched for PGF receptor alpha and SCA1, and these CAR cells that are enriched for CXCL12 and leptin receptor. Now, our initial hypothesis is going to be that these are actually going to be located in unique domains within the bone, and that their transcriptional profiles are simply reflecting their unique spatial environment. And so what we started to do is, through this iterative process, start to map these different cell types and see where they're present. And so kind of help orient you, we start with osteoblasts because we know where they should be. And as you look at these graphs, I want you to imagine as we move along the x-axis, it's how confident are we that a spatial spot is going to contain that cell type of interest. And as we move along the y-axis, it's how many of those spots do we have at that confidence level. And so you can see here that towards the end, there's a little bit of osteoblasts on the cortical bone, but the vast majority are present on the trabecular surface, which you know are more active anyways. So it seems to be working. We look at these CAR cells. Now we can see that they're heavily enriched within the bone marrow, where we'd expect our skeletal stem cells to be. But when we get to these P-alpha-S cells, we can see them heavily enriching in the cortical bone. And when we look back at our actual plots, it's not the cortical bone, but this outer cortical surface where there's a tissue known as the periosteum, which is known to contain its own unique skeletal stem cell. So before you think that I'm bashing on the people who did this work, you should know that I was the one that did the single cell analysis for this original paper. And the problem was that we went into it thinking every cell that we're collecting is from the bone marrow, and we have two populations that seem to be expressing stem cell markers. But what really happened is a little bit of contamination from this outer edge of the bone has gotten in there. And instead of us thinking that might be a possibility, we attribute it to, it has to be in the bone marrow because that's my experimental design. And so I think that spatial can also, in a way, be a bridge between a lot of the data that's already out there. And we can start to use this as a scaffold of saying, where are they located? Are they the unique same cell type, or are they actually different? Is it the same cell type but just transcriptionally different because they're in different regions? And so in this case, we can actually pick out some of the mistakes that we have made and suggest that actually some of these cells might be from the periosteal stem cell population. And so this was also, I think, very good confirmation that this predictive modeling, so this way of spatially deconvolving, can be used to pick out those spatial spots that are likely containing the real bone marrow-derived skeletal stem cell populations. But now that we have our spots in the bone marrow that have the stem cells, what other cell types are often present in those same exact regions? And so we started to do correlation analysis. We make a nice colorful dot plot. And if we put it beside our cartoon of the bone marrow niche, everything on the right are cell types that we think every time I find a skeletal stem cell in the bone marrow, I find these same cell types in the exact same spatial spot. And so we get smooth muscle cells, endothelial cells, macrophages, and to a lesser extent, megakaryocytes. And I picked this cartoon for a reason because lo and behold, they exactly match up with this, except this took days and this took decades, right? So while I would trust the work that went into this far more than I would ever trust these dot plots, this is again a way that we can start either developing hypotheses to focus our efforts, to validate these things. And you can imagine if there was some kind of perturbation, you could easily assess whether the stem cell niche composition could change using a method like this. And so we're shaving down and generating these ways of focusing in our efforts into something that may be more biologically interesting. But we can take this one step further. We can not just say that endothelial cells are present in this stem cell niche, but we can subcluster these endothelial cells and say, all right, each of those endothelial cell subtypes are localized to different regions in the bone. They have unique transcriptional profiles. And when I look at the same kind of correlation analysis, I can see it's not all endothelial cells, but this EC3 that's enriched for notch signaling that's actually present more so in this stem cell niche. And of course, we picked the notch signaling because this has already been known as something driving stem cell biology, is this endothelial cell to stem cell notch signaling. And you can do this for whatever cell type you want. We can look at macrophages. Again, we can find unique transcriptional profiles, unique positioning, and find out which of these subtypes tend to be more prominently expressed or more prominently present within the skeletal stem cell niche. So in this case, I think we took lemons and made lemonade, I would say, in that our spatial spots are big, but instead of thinking of them as that's not single cell resolution, let's think of them as niche spots. And so now we have basically each spot can contain the stem cell and all the other support cells that are going to be there. So the next question is, okay, we know where our stem cells are. We know what other cell types are present within that niche. The next thing we want to know is how are they talking to each other? And so when we do this from single cell, you're going to look at the cluster differentially expressed genes. You'll figure out which ones are secreted. You'll have ligand receptor pairs between different clusters. And then maybe you can run some packages like NicheNet or CellChat or CellPhoneDB or whatever you're into. And those are going to give you, we predict that cell type A will talk to cell type B via this ligand receptor pair. And we usually stop there, right? And the problem has always been that this is just a computational prediction, right? If these cell types never come anywhere near each other, then what it spits out is completely irrelevant, right? But if we can add this spatial information, now we can start to dissect down what the computer predicts as a potential way to communicate down to what cell types are actually close enough that they would actually use this method inside the animal. And so we can look to see which of those ligand receptor pairs are actually expressed by cell types that we've just identified being in the niche. And we can actually look within our spatial data and say which of these ligand and receptor pairs are actually expressed in our spatial data in that spot that has the stem cell. And so you slowly start with a giant list and whittle it down into fewer and fewer options until you end up with three basic groups. We had one group that we found to be spatially restricted. That means that it really is only expressed in our spatial data where we predict a skeletal stem cell to be present. But when we look at single cell, it's expressed by every single cell type that's present in that stem cell niche. Then you have patterns like this cell type restricted. So it was only expressed by a cell type that we had predicted to be in the stem cell niche in the single cell, but it's also expressed by that same cell type if you look outside the niche as well. And so in this case, it's a monocyte-derived factor that it doesn't matter if it's in the niche or not, it's going to express it regardless. Oh, sorry. And then the final one is a combination. It's only expressed by cells that are in the niche, and only when they're in the niche is that going to happen. And so we took a stab at doing RNA scope in bone marrow, and you can be the judge of how successful we were, but we saw these same patterns, right? The things that we were finding from our predictions tended to be more closely associated with these two cell types that are expressing this ligand and this receptor. And so the final idea I want to talk about is these idea of microdomains, or however you want to refer to them, but we know these exist, right? We know that the blood vessels are going to be releasing certain factors. They're going to affect the availability of oxygen, the availability of metabolites. We know in the context of bone that the growth plate is secreting factors as it enters the transition stage. We know that bone is releasing a bunch of growth factors, and it's also going to be dependent on forming and resorbing surfaces. So we know they exist, but it's really hard to address how are you going to pick out what genes are regulated along this environment, other than really staining and hoping for the best, right? And so we applied this same spatial time technique, and I think we can all agree that this looks like a hot mess. But what we've done is take every single bone marrow spot and color code them based on how far away they are from the nearest blood vessel. And again, we're not only doing it to the one sample, we're doing it to multiple samples, and now we're aligning every single marrow spot from multiple bone samples and saying, how far away am I from my tissue of interest? In this case, it's the blood vessels, but we did trabecular bone from cortical bone. Everything gets compiled and squeezed onto a linear axis. And so one of the benefits of this as well is we don't actually have to know what we're looking for when we do these kinds of analysis. So we steal the same people we stole the name from, we steal their programs as well. And so pseudotime will calculate a pseudotime value, and they will tell you every gene that changes as you move along this pseudotime axis. And what we go in is delete what they calculate for their pseudotime, and we ram in our distance calculation, and we say, do exactly what you're designed to do. And so now it's going to tell you every gene that changes in the bone marrow as you move away from, in this case, the blood vessel. And we can look and see, well, what genes are enriched right adjacent to the vessel? Which ones are enriched as you move away from them? And so we can start to generate hypotheses without really having any idea of what we have to look for. And you can start to see things like changes in metabolic genes, where glycolysis is favored in the stem cells near the blood vessels, and then oxidative phosphorylation takes over as you move away. You can look at signaling gradients for TGF-beta and Wnt and PDGF, and not just that, but you can look at how far their influence extends. And for instance, for Wnt signaling, I don't know if you can see, but it's very high near blood vessels and it drops down very quickly. And so again, this has been painstakingly dissected to show that Wnt ligands really don't travel very far. The zone of action from their source is very confined. Whereas things like PDGF signaling, it's known that these affect a larger area. So not only can we identify pathways that are changing in an unbiased fashion, but we can also get lucky and find that these actually reflect very well in terms of the distance of impact from the ligand source. And again, we can do some immunofluorescence and we can confirm some of these findings. So in this case, we're using spatial transcriptomics to unbiasedly identify and interrogate some of these pathways that we really never knew to look for, and to piece together multiple samples to give us an average gene expression as we move away from the bone surface, from the blood vessels, whatever your tissue of interest is. And so all of this was in the context of a normal, healthy bone. Now we want to start looking what happens if we do some kind of perturbation. And for this, we joined up with Jonathan Rios from Scottish Rite, and he works on a disease called neurofibromatosis type 1. And this is a mutation in the NF1 gene that leads to aberrant RAS activation. And it's a pediatric disease that has a whole plethora of disorders associated with it, but one of them is these skeletal problems. And the main manifestation is this bowing of the tibia. And so you'll frequently see these children will have in utero fractures, and as they develop, they'll continue to fracture regularly. And not only is it a bowing problem that causes mechanical issues, but when they fracture, they heal very poorly and they form these pseudarthroses. Instead of a nice healthy callus, they'll go on to heal. And so unfortunately, they heal with such, or they injure with such frequency that oftentimes the treatment is to do a lower limb amputation and fit with a prosthetic. Otherwise, they get so far delayed in terms of their ability to walk. So the current therapy that they use at the pediatric hospital is addition of BMP ligand on a sponge along with some fixation. But there's a mixed bag of whether they think that's actually positive or not. And in fact, the most recent study came out saying it may actually be deleterious. And so either way, they don't really have a great way to go in and try and help these patients to heal these recurring fractures. So we came back and developed a mouse model. We ran spatial transcriptomics on this tibial fracture. This is 10 days out. And again, go through the standard pipeline of quality control. How many genes are you expressing? We use transcriptional information to kind of annotate which clusters are which. But honestly, from the morphological data, it's not hard to tell. You have the fracture line that has a little fibrosis. You've got cartilage that's forming to try and stabilize these bones, and eventually it'll transition into woven bone. We can use marker genes to make sure that everything's expressed where you'd think it would be, and we can plot some UMAPs to say that indeed bone expresses bone-related genes and cartilage is more cartilage-related transcripts. We can start to look at these transition zones. In this case, on the left, this is a general cartilage marker transitioning into green, which is more a prehypertrophic, hypertrophic chondrocyte, and then eventually transitioning into blue, which is where your woven bone is going to form. Or we can do groups of genes all linked to BMP and TGF-beta signaling that's driving cartilage down the center and then bone formation along the edges. And we can once again apply our spatial time technique and see the exact same thing. This transition of basically healthy healing of transition from early to late cartilage and then early to late bone. We can then again go in and unbiasedly start to say what types of gene programs are activated right at the injury site, and as we move along this kind of stepwards progress, and we can do pathway analysis to find out indeed it's basically a healthy healing fracture. So to mimic this pseudoarthrosis NF1 patient, we knocked out NF1 using periosteal Cree. You can see normally it's highly enriched along this outer periosteal surface, and when we fracture these mice, it'll expand to label a lot of cells that will comprise this fractured callus. And when we look in our spatial data of the two, you can see that we're preferentially targeting cartilage, a little bit in the ossifying perichondrium and some woven bone, but most of the clusters seem fine. Now, the result of this in the mouse model is that while at the top you can see this cartilage in blue transitioning to bone and fibrotic tissue on the top, this NF1 mutant mouse basically has low levels of chondrogenesis, but a lot of fibrosis, and it never really makes that transition from a soft, squishy callus made of cartilage into this bony bridging that we really need to facilitate healing. And so again, when you look along these fracture lines, so the fracture line is going to be on the left, moving more distant. In black is our wild type. And you can see this wave of BMP activation that happens as you transition from cartilage to bone, with a little spike in MAP kinasing right around this transitional region. In comparison, these NF1 mutant mice in red, you can see absolutely no BMP activation across the entire callus. And this is associated with elevated levels of MAP kinase, again, throughout the entire tissue. And so this is and we also stain to validate using phospho-SMAD staining. And so this is interesting in the context that we know that MAP kinase can actually drive expression and production of BMP inhibitors. And so you can imagine that if this hyperactivation of MAP kinase is going to drive BMP inhibitors, which is going to make it less responsive to any kind of outside source of BMP ligands. And this might help us explain why these patients are not really responding to the treatment like we'd expect. So right now at Scottish Rite, they are actually doing a combination of MEK inhibitor and BMP ligand to try to overcome this. And so far in their limited phase clinical trials, it seems to be having promising results. So we also tried to run spatial transcriptomics on an actual human sample. So this is from the pseudoarthrosis site of an NF1 patient. And you can clearly see this kind of enrichment of fibrocartilage kind of down the middle with some bony healing on the ends. But it's important to note, while all the mouse work I was showing you was from 10 days post-fracture, so it's kind of transitioning from cartilage to bone. In this is a patient who fractured a year ago. And so this is not some kind of transitional state, and if we just give it some time, I'm sure the cartilage will resolve. These patients are stuck in this stage with this fibrocartilage that's just sitting there. And as long as it's there, it can never kind of transition over and to start to heal this bony gap. And so we did spatial, and we started to look at some of the same pathways, and we did our spatial time, and consistent with what we were seeing in the mouse model, we see hyperactivation of MAP kinase right within this kind of pseudoarthrosis site. We see relatively normal TGF-beta signaling, but a complete absence of BMP activation, unfortunately. And again, you can see this with our phospho-SMAD, although this one's not as dark, but you get some along these bony surface, but within this pseudoarthrosis region, really BMP activation is lacking. And so this data was suggesting that NF1 deficiency is leading to impaired bone regeneration, and potentially through this hyperactivation of MAP kinase and this inhibiting of BMP signaling. So where are we going next? We talked about fracture healing. I talked about the limited mammalian regeneration, but I told you we were going to get to axolotls, and here we are. So I'm not too sure how we ended up here, but someone decided that we need to start working with some weird organisms, and axolotl definitely fits the bill where, like I said, you can cut out the whole limb, you can cut out parts of the heart, parts of the central nervous system, and somehow they will regenerate it just fine. And so in this case, if you give it enough time, this entire limb will regenerate. But it's always been an interest of, how is it able to do this, right? How come they get a whole arm and we can barely get a fingertip? Right? And so there's obviously been a lot of work done, and this is collaboration with Kate McCusker in Boston. And one of the things that they focus on is this idea of patterning. So you're not just going to randomly grow an arm. You're not going to grow a finger coming out of your elbow. That doesn't make a lot of sense. You need something that patterns it back to the way it should be. And one of the things they know drives this is anterior and posterior patterning. And so if you do not have tissue from both anterior and posterior derived sources, these blastema that form after you injure will kind of grow, and then they'll shrink back in and regress. And so you need to have signaling components from both of these two tissue types in order to facilitate limb growth. Now, the classical model, quite straightforward. You draw a line right down the middle, and you've got anterior tissue on one side and posterior tissue on the other. And they've worked out some of the signaling cascades, like Sonic Hedgehog and FGF, basically feeding each other in a loop to basically help say, okay, you're on one side, I'm on the other. The problem became, as you amputate these axolotls at different planes along your arm, they always seem to know exactly what needs to be regrown. So if I cut off the hand, they'll regrow a hand. If I cut off the lower arm, they'll regrow a lower arm and a hand. And if you go at the shoulder, they'll go upper arm, lower arm, and a hand. So somehow they know exactly what needs to be done. And when you start looking at the blastema that are forming at these different amputation planes, you actually start to see changing abundance of anterior and posterior gene expression patterns. And so this led her to postulate this border shift model, where instead of drawing it right down the line, all we're going to do is rotate it a couple degrees. And I have to admit, that didn't sound super exciting to me either. When I first saw it, I said, great, we're going to draw the line at a slightly different angle. But now you can imagine that this is going to have huge implications in terms of knowledge of what's going to be regrown. And so in science that I'm not sure how gets funded, they can show when you start playing around with these anterior or posterior programming, you can regrow, you can amputate at the wrist and make them regrow a whole nother arm. And unpublished data, she's shown me that they can do this multiple times until you end up with this mega arm growing off of it, right? Again, I don't know how that gets published, but this moral of the story is by simply slanting this anterior-posterior boundary, now at every stage along this arm, we have a different abundance of each of these two populations. And this will obviously give you this intrinsic knowledge of where am I along my proximal distal axis, right? By just tweaking this a few degrees, now we have a way of saying where are we in the arm and what needs to be regrown. Is it going to be that simple? Probably not, but I think this is at least one contributing factor to it. And so when we start looking at spatial of a regenerating axolotl limb, and we look, so the y-axis on this is essentially how good are we at distinguishing expression of anterior and posterior genes. And this is ranked in terms of, I don't know, this is the new stuff that just came out. It's ranked in terms of the overall ability across all genes. So it's going to be normalized in a slightly different way. I'll show it to you in a more clear way in a little second. But what you can see is that as you slowly rotate this line from going straight down the arm, slightly at an angle, you can see that now our ability to distinguish anterior and posterior genes are going to slowly increase. And if we rotate it too far, now we're starting to really separate proximal and distal, and now it drops off again, right? So I told you there's an easier way to look at that, and that is by just simply graphing how good are we, or how significant is this expression of anterior and posterior genes along this axis. The classical one we set to 1, and the border shift model now jumps to around 3.5. So we're showing a significant improvement in how well we're able to distinguish anterior and posterior genes. But I hear what you're all saying, I don't really care about axolotls, and I understand that, even though they're super cute. But don't worry, there's something you do care about. The exact same thing is seen in human limb development. So when we do spatial on these fetal limbs, and we again slightly rotate this axis from going right down the middle to a few degrees off center, now again, we're able to vastly improve our segregation of anterior and posterior tissue, while at the same time, proximal distal genes really aren't as affected. And so we think that this is going to be kind of a universal way that limbs are going to be developed, regenerated, where you are going to have these altering ratios of anterior and posterior tissue. And we actually have new work that's saying that the relative abundance of these anterior and posterior genes can actually drive some of the genes known to label proximal and distal regions as well. So I think they're all going to be interconnected in a way. And so with that, I'd like to thank the lab, the funding sources, the collaborators that I've been working with throughout. Each one of them pretty much represents one of the projects that you saw today. Thank you for the invitation, and I'm happy to take any questions.
curious about this balance between AMP signaling and the fibrotic programs in the mutant mouse you guys made. So it sounds like you guys are targeting MAPK inhibition to block BMP inhibitors. But what's the opposite side of the coin with regards to agonists of TGF-beta signaling, agonists of the Notch Wnt pathways, pro-fibrotic pathways?
So we haven't really, I don't think we noticed any changes in Notch. I think TGF-beta is slightly altered, but not nearly as much as BMP. And I think some of the in vitro work that was done earlier was suggesting that it's really an issue of like that terminal osteogenic differentiation. So this is why we focus on BMP. But yeah, it's kind of like a snapshot of like a highlight reel, right? There's definitely a lot more pathways that are going to change. How the MAP kinase ties in, or if it's some other component of this RAS GTP signaling cascade, yeah, I don't really know. So, but there's definitely a huge team at Scottish Rite that they're really focused on this area of research. We just kind of hopped in for the one study, so I'm pretty sure there's more information out there than I'm giving you.
So did you use the cryostat to actually section your samples, and what about the thickness? Because if it went too thick, would it cause any eye diffusion?
Yeah, and you have multiple layers of cells as well, right? So the question's about, yeah, how are the samples prepared and sectioned? So the calvaria and the digit tip, these were fresh frozen in OCT and sectioned just as they are. And so by playing around with what blade we're using, what temperature we're cutting at, things like that, we were able to get sections through it because we're talking about a P0 calvaria, so it's a little mineralized, but not so much. And these digit tip bones are very small as well. We're not going to win any awards for histology, but it was good enough to get forward. So things I can suggest, we noticed that including some surrounding soft tissue can help. And so in the context of the calvaria, the first try, we kind of like trimmed a little slot right down there, and the calvarial bones started to kind of like wave at us as they peeled off the slide. So by adding a little bit of like the surrounding muscle tissue, or in long bones, the same idea, this actually helped kind of adhere to the slide better. We often section our samples so that the ends are sticking outside of the area of interest. And that's because if you're going to have tissue detachment, then you want to have it where you don't actually care. And the thickness of those were all 14 microns. So I would say that it's very likely that there's multiple cell layers on top. The good news is both the suture and the digit blastema, it's kind of the same cell types underneath, I guess, is what's visible. If you started to get into something like bone marrow, where it's like you can have different cell types on top of each other, it'll be more challenging. Everything else was this formalin-fixed paraffin-embedded samples, and they're sectioned at 5 microns. So I don't think you have this issue about overlapping cells. And I was talking to a few people. It's an ongoing battle to like decalcify it while at the same time maintaining RNA quality. And so there's definitely some tricks we've learned, cardiac perfusion when possible. We do everything under vacuum in the fridge to help like reagents get penetrated in. We've had good success with like a EDTA with formalin mixed in. We've used some special reagents from ACD Bioscience. It really depends a little bit on your tissue. Like we've tried to do this on human femoral neck fractures as well. And there, if you do an acid-based decal, the outside is burnt by the time you decalcify the center. And so we choose other methods that'll kind of slowly decalcify while still preserving the RNA. Does that kind of answer your questions?
Actually, the capture region on site is very small.
It is.
Your sample is kind of long, maybe inside. So, like, which section would you choose?
So I guess that'll come down to what's your question of interest, right? So in this case, it was half a mouse femur. A lot of the human bone samples we'll try to trim down to be somewhat in there, but we're interested right on the fracture line. So they do have these 11 by 11 millimeter reaction areas. I mean, yeah, you can do as much as you want if you have an infinite funds, right? But they do have other ones for like larger, like precious samples that you don't want to trim down. But it's really going to come down to, yeah, where are you most interested in? And we always do like multiple repetitions of it to kind of like, again, build the stats. Like you have to kind of think of it as doing single cell. You wouldn't want to do analysis with like 50 cells, right? You're not going to get any information. So it is a little bit of a guessing game about how many reactions do I need to run before I'm going to get critical mass. Young tissue is better, injured tissue is better, because they just tend to be more transcriptionally active. Aging, quiescent tissues, you're going to get less, right? So, but I have no magic rule that'll solve all your problems, unfortunately. Yep.
Hi there. Yeah, thank you for your talk. I was wondering if in the digit tip regeneration model, if you change the angle of amputation, if you can change like the length of grow back as you see like kind of in the X model.
So I think that if you amputate less, so if you amputate more distally, this histolytic event actually extends further. And so for whatever reason, the less you trim, the more it trims itself. Honestly, they're not entirely sure why it does this. So you'll have this osteoclast-mediated activity that will cut off a piece of bone, and it actually, we call it ejection. It's not shooting out, but it'll slowly get pushed out, and then the wound epithelium will close in behind it. You can block that completely, and they'll still regenerate just fine. So it's not essential. Some ideas was you have to like open up the marrow cavity to allow the release of stem cells from there. But again, like you can block it and it's still fine. So they're not entirely sure why, but yeah, but you can move the amputation plane around, but it'll still end up kind of deciding where it wants to regrow from up until you start messing with the nail bed. So there's a few major signaling hubs in there. One of them's the bone stump, one of them's the nail bed, and one of them's the wound epithelium. And if you start to disrupt any one of these three, the whole regenerative process kind of collapses.
Rob, I have something from the chat. Sean Hallett says, try Morse's solution, 24 to 48 hours to preserve RNA in calcified tissues. He's had success at P21, P49, and cranial base and long bones. I don't know what Morse's solution is.
Hey, I don't know this either.
SES, so try that.
Okay.
And then Andreas Apsalis asks, in your 55 micron Visium slides, did you consider adopting Bayes space for subspot probabilistic clustering?
Holy smokes. Sounds like you know more than I do about it. So why don't we go back to the beginning? Hi, I'm Robert. I'm a cell biologist. Self-taught in bioinformatics. So I'll say that a lot of these packages they're using, they're not just designed to say what's the probability of this cell type being present in my spatial spot. Some of them actually use the neighboring spot information to say it's actually going to be more on this side or this side. I think that's probably what he's trying to get at, is trying to not just say within this spot where are you, or like who's there, but where are you within this single 55 micron spot, if I'm understanding correctly. And so some of them are designed to do that, but honestly, I think the limitation was the quality of our data, that we could have done that, and it
He says that's correct. So nice job.
Boom. So yeah, cell to location does that for you, but I mean, I wouldn't believe it even if we did, right? At some point, your data reaches the limits of what you should believe. And I think maybe this is something we all do a little bit of, like, I'm just going to do a little more analyses. I'm going to make one more plot. And you can make it look great, like look at my huge fold change, but in reality, it's meaningless, right? I went from 1 transcript to 2 transcripts. I mean, you're not learning anything from that, right? So I would say we were on that edge where our data quality was not sufficient to really make any meaningful comment about it.
Excellent presentation. Question I have for your special, you use the formalin fixed tissue, would you compare with frozen section, which is the most of the time, which is more special?
So I mean, we've never done fresh frozen and FFPE on the same tissue type. So we're actually running another round of digitip spatial now, and so then we could probably compare. But yeah, we've never done this before, so I can't really say which one's better than the other.
FFP is very easy, it's a lot of
Yeah.
tissue is already in formalin, but can do that one.
So I should also say, like, I mentioned a little bit sometimes about how this was like version 1, and so it started off where you put your tissue directly onto their slides. Then they've gotten to now you section onto another slide and you transfer using their expensive panini press, right? So it used to be with fresh frozen that you would have to enzymatically poke holes in the tissue and allow the mRNA to physically leak out. And there was another test you would have to do that says if you don't poke enough holes, no mRNA is going to come out. And if you poke too many holes, it's going to just go everywhere. And so you would have to find kind of the sweet spot of how much do I need to digest for it. Now I think they're moving even the fresh frozen over to this library of probes that they send in because the kinetics are according to them and probably in reality are far superior, right? So now you don't actually have to pull the mRNA out. These little oligos go in, they bind, you rinse away whatever, these oligos come out and bind to the surface. So I would imagine that the gap is closing between these two different like sample embedding. I think for us it was you can't section adult bone without decalcifying, so we had to do it. I think if it's soft tissue, if it's in, depending on where it's coming from, sometimes it's only available to get in one way or the other. I feel like I would probably err on the side of FFPE, not just because it's more common, but I think it's easier to work with. The samples we're doing in FFPE is 5 microns or 4 microns sometimes, but the fresh frozen was 14. And I would say the amount of information is relatively comparable. So I would guess that it's probably just more efficient to do FFPE. The sectioning looks nicer, so.
Okay, any more questions at all? If not, we sort of hit the hour, so for being here, appreciate it.